Hi, everyone. This is part 17.4 of chapter 17. We're going to be talking about epigenetics. This first one will just be an overview, and then we'll go into a little bit more detail looking at epigenetic changes that are either programmed during developmental stages or are caused by environmental agents. So first, we're going to talk about what we mean by epigenetics and epigenetic inheritance. And the main thing that you want to think about is that this means that the sequence of the DNA does not change, but there is some influence on the actual gene expression that in some cases can be inherited and will change basically or cause gene regulation of particular genes. So we'll talk a little bit about that in this particular part of the chapter. So epigenetic regulation, we know that this goes on, and the term was actually coined in 1941, and it's been used to describe this very wide variety of things that we see with the expression of genes. So we want to think about it first of all as a change in gene expression, meaning the rate of transcription of a particular gene that is not based on the DNA sequence itself. So it's something on top of the DNA sequence itself. There can be long-term maintenance of the change in expression, meaning that it can be passed from cell to cell, and in some cases it may be passed from generation to generation, even though it's not the DNA sequence itself. So the definition for epigenetics is that it's the study of mechanisms that lead to changes in gene expression, which can be passed from cell to cell, are reversible, which is different than in general that we think of changes to DNA sequence, and do not change the DNA sequence. So the actual genetic sequence remains the same. It's actually just the transcription level of it or the expression of it that changes. So some of the ways that we've already talked about this were earlier in this chapter, and we'll kind of do a quick, maybe brief little overview of a couple of these again. But in general, we think of some of the different molecular changes would be DNA methylation. Remember, we talk about that. Generally, we think of DNA methylation as downregulating transcription. We talked about chromatin remodeling and covalent histone modification. In general, we tend to think about this as well as the localization of histone variants as upregulation of transcription. And then we talked some about feedback loops, meaning that if a lot of something is being produced, it may do a feedback to stop the transcription. So this table just basically does a quick summary of these. If you need more information, most of this was from earlier in the earlier in the chapter. But just some quick kind of refresher talking about some of this epigenetic modification we might see by a transcription factor. In this first example, it's showing that we have a specific gene. Remember, we see no changes to the sequence, but that a transcription factor may recognize a specific gene sequence and bind to it. This transcription factor then has the abilities to recruit other proteins that may come along that could do histone modifying, or in this case, DNA methyltransferase, where methylation ends up happening. And in general, we would typically think of this as down-regulating that particular gene. We can also have where we have a specific gene and we have a non-coding RNA that recognizes that specific gene sequence and binds to it. Remember, we can have these as complementary. And when this happens, it again can bring in other proteins. And in this case, it's showing that methylation could occur. Again, generally speaking, we'd think of that as a down regulation. Epigenetic regulation, though, can also happen in two kind of bigger ways that we're going to talk about. And this can either be developmentally or environmentally. So many of the epigenetic modifications are just normal part of development. For example, one of those that is a really good example is what we call genomic imprinting. And what that means is that one particular allele, in the case of IGF, IGF2, this one, the maternal allele is always silenced and only the paternal allele is active. And this would continue generation after generation and what we would consider in a developmental way. Another one that we see in females and humans is what we call the X chromosome inactivation. Males are born with an XY, an X chromosome and a Y chromosome. Females generally we think of as born with an X X chromosome in order to not get overexpression of the genes that are on the X chromosome. One of the X chromosomes will become a bar body. And in this case, it means that it becomes transcriptionally inactive. It basically condenses down to where none of the genes on it are expressed. So only one of the two X chromosomes would be expressing the genes. In addition, we can see that environmental agents can also affect epigenetic regulation. This could be things such as temperature, diet, or toxins, and we'll look at an example later in this chapter on this. So this is just a quick one to talk to summarize what I was talking about. The thought of program changes during development. Again, we can have these programmed ones that we see during the synthesis during oogenesis and spermatogenesis. And in this case, there's patterns that would affect the maternal or paternal allele being expressed in offspring. The X chromosome inactivation, I mentioned as well, that one of the X chromosomes would be inactivated during embryogenesis in female mammals, and that's so we don't get overexpression of those genes. And then cell differentiation. So you want to think about embryonically as cells begin to differentiate, we would see that there would be epigenetic changes so that there is DNA methylation or covalent histone modification, so that in different tissue types or different cell types, we would see different expression of the genes. We'll also look a little bit at environmental agents. Let me see if I can make this a little bit bigger. Not sure why that's cutting off on that one. Sorry. You may have to look in your book on this, but we can look at temperature. This is common in some plants. That cold weather temperatures would cause specific modifications that affect expression, and then the following spring it may actually change. We can also see this in diet. We'll look both at bees, we see this, and we also see this in some cases in mice. And then also with toxins, we can see that cigarette smoke is one that we see epigenetic changes in lung tissue. So we can see increase of lung cancer because of it. And that's the end of this particular small section. That was just kind of the overview. And then I'll move into our next one. We'll be looking at epigenetics and development. So I'll talk about that in our next mini lecture. Please let me know if you have any questions.